all, very honored to be here, and thank you so much for inducting me as a fellow of the Academy. And it's like, thanks for giving me the opportunity to present my work here. So now, from the previous talk where the speaker talked about classifying the clouds, I'm moving to this topic where we are going to molecularly classify the prostate cancer. So the disease I'm going to talk about is a male-specific gland, prostate gland, and the main function is to secrete seminal fluid, which nourishes the sperm. And basically, anatomically, this gland is divided into 4 different zones. And if you look, these zones, they are very specific, and the propensity to develop cancer is totally dependent on where it's located, which zone is like more have like more potential to develop cancer. And upon enormous like studies, like histopathological studies, people have found that peripheral zone is more prone to develop cancer, followed by transition zone. And looking at like much closer at the histopathological grading, this is a foci. There are millions such foci in gland, and if you look at these foci closely, there are three different kind of cells. There are like basal cells lying along the basal lamina. There are luminal cells which are lying along the lumen, and then there are also few neuroendocrine cells. And because of the neoplastic transition here, like these cells, luminal cells, they start dividing, they start proliferating, and as a result of that they form adenocarcinoma. And usually, since prostate cancer is very much dependent on androgen signaling, these patients, they receive androgen deprivation therapy, and over a period of time, these patients develop resistance to this therapy, and they progress as a castration resistant adenocarcinoma. Then from here again, due to like de novo or maybe as a resistance to endoandrogen therapy, these patients develop neuroendocrine cancer, and which is like really challenging to treat. So if you look at the molecular alteration, there are so many. So I'm not going to go in detail of these alterations, but just want to give you an overview. So when we look at the molecular level of this cancer, see, one of the most recurrent, highly recurrent subtype is TMPRSS2 ERK genetic rearrangement. So here TMPRSS2 is androgen driven gene, and where is aberrantly get just exposed to ERK. And as a result of that, this ERK, which is not expressed in the gland, is now highly expressed and is recurrent in 50 to 60% of the patients. Whereas RAF kinase rearrangement is only present in 2% of the cases. SPINK1, which does not have any genetic rearrangement, is highly expressed in 10 to 15% of the cases. And these cases which have like a higher expression of SPINK1, they are associated to be with the aggressive subtype of disease. They have a higher Gleason score, which is a way of grading the cancer, and have shorter biochemical recurrence. Now, when I started my own group in 2013, that time one of the challenge was when you look into the literature, there was no such reports like what different subtypes of prevalent in Indian population. So what I did, like I collaborated with different clinical institution in the northern part of the India, collected some sample which have been collected by different means like prostatectomy where the entire gland is reduced, or needle core biopsy as shown here. There are like usually 12 core needle core biopsy, or truss, which is a method to resect that gland. So what we use some of the very traditional method like immunohistochemistry, RNA in situ hybridization, and FISH. So you can see here, this particular patient is positive for TMPRSS2 ERK genetic rearrangement, and this brown stain here is showing overexpression of ERK oncoprotein. But then looking at the genetic level, this FISH probe here, this is the intact locus where there is no genetic rearrangement, and here this probe is bake apart probe. If there is genetic rearrangement, you will see 2 distinct color probes like red and green. So this particular patient is having a TMPRSS2 ERK genetic rearrangement, whereas in case of a SPINK positive patient, there is no genetic rearrangement, but you see brown staining, very peculiar brown staining in this particular patient. So when we summarize data, we found still in northern part of the India, like 49% of the patient, they harbor this TMPRSS2 ERK genetic rearrangement. SPINK1, which is similar to other Caucasian cohorts. RAF genetic rearrangement, which is 2% in Caucasian, was 6% in northern part of the India. And good news was this, like these patients could benefit by the RAF kinase inhibitor, which have been used for melanoma treatment. Now, Vipul, my PhD student, that time, like when he joined my lab, so the question was like, if SPINK is not having any genetic rearrangement, what is the mechanism which is underlying this SPINK1 overexpression? So we narrowed down like to 2 approaches, post-transcription regulation or transcription regulation. So he looked into the post-transcriptional mechanism, and we narrowed down 2 microRNAs, 338 and 421. And I'm not going to go into the detail how we narrowed down, but we looked into these patients, like categorized into 2 groups, SPINK1 positive and ERK positive, fusion positive, and we found, we found that the SPINK positive patient don't show any expression of these microRNA. So the question was like, what is the mechanism? But then before we move on to that, we thought like, what about if we replenish this microRNA in a SPINK positive prostate cancer cell line? And we found there was a decrease in oncogenic potential of these cells when we overexpress this microRNA. You can see here, this is a animal xenograph model where we implanted this is SPINK1 positive cells which were overexpressing either these 2 microRNA or just control. And overexpression of these microRNA results in slower growth of tumor in these immunodeficient mice, which indeed shows that there is a decrease in oncogenic potential. And again, when we looked into perform several oncogenic assays, cell-based assays, and we found there was a significant decrease in the EMT and stemness, and probably the reason was we found that these 2 microRNA, 421 and 338, they do show binding on the 3' UTR of multiple genes such as like ABCG2, c-KIT, and several other genes which play critical role in EMT and stemness. Now looking into the mechanism, why these 2 microRNA are silent in or are not expressed in SPINK positive subtype. So we thought it could be either deletion of this microRNA locus or epigenetic silences. So these we examined by doing genomic PCR, these locus were not deleted in SPINK1 positive patients. So we thought it could be epigenetic mechanism, and what we did, we stained this patient tumor microarray which has like multiple tumor biopsy from multiple patients. So it has like around 237 small biopsies from prostate cancer patient, and you can see most of the SPINK1 positive patient, they show higher expression of EZH2. Around 88% SPINK1 positive patient, they show higher expression. So we thought since EZH2 is a methyltransferase, is it like playing critical role in epigenetic silencing of these 2 microRNAs? So for that we designed an experiment, and we use these SPINK1 positive cells where we overexpress 338 and 421 microRNA, and we found that indeed in our CHIP PCR data that EZH2 show very prominent recruitment on the locus of both microRNA, which is also accompanied with H3K27 trimethylation mark, which is supposed to be coming from epigenetic silencing through EZH2. Now, so this is one story from the lab, and then we kind of showed like if we use epigenetic drug, we could revert this condition. We can make SPINK1 go down because overexpression of these microRNA negatively regulate the expression of SPINK1. So now I'll switch my gears towards androgen deprivation therapy, and this gentleman here, Charles Higgins, he was the first one along with his collaborator to show that bilateral orchectomy, which is surgical castration of men, result in reduced tumor burden in most of the prostate cancer patient. And this was the study that they published in 1941, and later he got Nobel Prize in Physiology in 1966 for this study. And from since this time, like a lot of pharma industry, they started investing in developing anti-androgen therapy drugs. It could be like either the biosynthesis level, because there are 2 organs which synthesize androgen, either adrenal gland or testis. So they use like abiraterone, or it could be AR antagonists such as biglutamide or enzalutamide, which will compete with the AR, which is a again ligand dependent transcription factor. So from here, like So these 2 students like Ritika and Nishad, they like dwell into this mechanism, and they want to see like what is the crosstalk between androgen signaling and SPINK1. Are there any transcriptional regulation in this subtype? So again, we went back to our tissue microarray specimens, and we stained for SPINK1 and androgen receptor. This is the brown staining you see. So there was an inverse correlation. So SPINK1 positive patient, they show lower or negative expression of AR, whereas a SPINK1 negative patient, they show higher expression of AR. So this is the combined data. You see SPINK1 positive patient 51, they show more high to more low or negative expression of AR, whereas like SPINK negative, they show more high to median. So which indicate that SPINK1 shows inverse correlation. So maybe androgen signaling is having some negative association with this particular subtype. So we thought like since AR is a ligand dependent transcription factor, is it that it's binding on the SPINK1 promoter region? We scanned the promoter and we found 3 ARE binding sites. So again we went back to our assay CHIP PCR, and we found when we stimulate this fusion specific, especially like these cells are androgen responsive with the synthetic androgen R188one, we see increased recruitment of AR on the promoter region of the SPINK1, whereas when we treated this cell with enzalutamide, which is an anti-androgen, we see decrease in the recruitment of this particular like AR on these sites. And likewise, we also see decrease in the mark of activation mark, that is H3K9 acetylation, which indicates probably like recruitment of AR is negatively regulating SPINK1. So, to recapitulate the entire thing in a cell line model, we chose LNCAP cells which are androgen responsive, grew these cells in androgen deprived media for a period of like almost 30 days. You can see there is a change in the morphology. These are the healthy cells, and over this androgen deprived condition, they start transforming into more neuroid like. So, there are like long neuroid projections. And when we sampled these cells at different time points and looked for the expression of different proteins, we found that we call this LNCAP as LNCAP AI, androgen deprived or androgen independent. There is a significant increase in the SPINK1 protein, whereas there is a decrease in the androgen driven PSA, which is one of the marker for prostate cancer. And not only that, some of the neuroendocrine markers such as synaptophysin, NOLS2 was highly expressed. Then what was striking was SOX2, which is supposed to be like a stimulus transcription factor, was upregulated, and we later on shown by doing several assays and also like some of the chip PCR that SOX2 play critical role in the transcriptional activation of SPINK1, whereas REST partner with AR and suppress the SPINK1. That's why it's coming up. So, to recapitulate this entire thing in animal model, we chose again immunodeficient mice, implant this VCAP cell orthotopically, and then once the tumor developed and the PSA level in the mouse blood was more than 5 microgram, we performed surgical castration and divided mice into 3 groups. So, one group was vehicle, and then they were treated with enzalutamide or another new generation anti-androgen that is apalutamide. And at the end of the study, after 4 weeks, these mice were euthanized and the tumor were collected, and the tumor was stained. So, you can see, you appreciate here over a period of time with these drugs enzalutamide and apalutamide, you see increase in SPINK1 followed by all these epigenetic neuroendocrine markers and including vimentin, which indicate that over a period of time when you put this anti-androgen drug in these mice, there is a resistance mechanism and these markers are coming up. So, this is the model that we propose, that when the patient they receive ADT, they go to CRPC, and eventually some of these patients they also develop NEPC, which is marked by increase in the neuronal markers. Especially we have shown that SPINK1 is upregulated, which is one of the important gene or important protein which maintain the neuroendocrine phenotype. And what we propose is like before we administer these prostate cancer patient with AR targeted anti-androgen therapy, it's important to weigh the benefits and the risk. And we also provide the paradoxical clinical the explanation or our way of explaining why majority of the patient who get this ADT, they develop resistance and they come up with a much more aggressive phenotype is because of this overexpression of SPINK1 along with other neuroendocrine markers. So, now I will switch to next story, that is DLX1, and the reason we chose DLX1 because we know like most of the cancer cell or cancer in general take cues or adapt molecular mechanisms which are involved in embryogenesis, and this particular gene DLX1, which is distillase homeobox 1, play critical role in the development of craniofacial features, jaw, and also in GABAergic interneuron. And not only that, in past 1 decade this DLX1 has been used as a urine based diagnostic marker for prostate cancer patient, not in India but in many western countries. So, we thought like first to screen what is a level of DLX1 in prostate cancer patient, and we found in the tumor microarray around 60% of the prostate cancer patient they show high to low expression of DLX1. Then the question was like why, like what is the correlation with ERG, because I showed you data where we have found around 50% of the patient they have ERG fusion status. So, when we look the same array stained for ERG staining here, we found like majority of the ERG positive patient they have high and medium expression of DLX1, whereas like ERG negative patient they do not show high or medium, it is mainly low or negative. So, 96% of the ERG positive patient they also tend to have DLX1, but not all the DLX1 patient have ERG expression. So then to understand the relevance of DLX1 in tumorigenesis or in carcinogenesis, where again performed a lot of like cellular based assays, but then I am going to only share the important mice experiment data here. So what we did, we performed CRISPR-Cas based knockout of DLX1 in these 2-RE1 cells and implanted these cells in immunodeficient mice, and you can see over a period of time there is a significant regression in their tumor burden in these DLX1 knockout mice as compared to the scramble control. Not only that, when we collected bone marrow and lungs after euthanizing these mice and perform allospecific PCR to detect the cancer cells in the mice, we found that the mice which were implanted with DLX1 knockout cell, they show less number of cells metastasizing to bone or lungs. Now, knowing that majority of the cancer patient, they develop bone metastasis over a period of time. So, the question was like, does DLX1 play any role in bone metastasis? For that, we developed this experimental bone metastasis model where we implanted either control scrambled cells or DLX1 knockout cell in the tibia region of the mice and let them grow over a period of time, like almost for a month. And then at the end of the study, we excise these bones and perform micro CT and then analyze the data by using bone morphometric analysis like software CTN. And what we found that majority of the all the mice which were implanted with DLX1 knockout cells, they show higher bone volume, bone surface area was also high, and also like the trabecular length was also high in the DLX1 knockout mice. And so again we kind of showed here that higher DLX1 expression is playing critical role in bone loss and destruction of the bone architecture as seen in scramble control mice. Now, to figure out what is the mechanism in this like in entire circulatory, again because majority of the DLX1 positive patient, they show ERG expression. So, we hypothesize maybe it's like a looping between the AR and ERG, because these both AR and ERG are known to co-regulate multiple genes, especially androgen driven gene, which play critical role in prostate cancer. We performed this sequential chip assay and we found immune complex which are collected by ERG, again followed by AR antibody, show enhanced enrichment of DLX1 on the enhanced enrichment of ERG and AR on the DLX1 promoter region, and this was used as a control. Again, this was validated by analyzing a publicly available chia-PET data, which is chromatin interaction assay, by using pattern tagging. So, you can appreciate here when we use this RNA-Pol chia-PET data and integrated with the ChIP-seq data for ERG and AR and FOX7, which are known to be a regulator for DLX1, there is a very starking looping between the ERG and AR, which is facilitating the transcriptional activation of DLX1 in DLX1 positive patients. So, now in the field it has been known that bromodomain inhibitor could work when for the genes which are driven by androgen signaling, such as because these BRD 2, 3 and 4, they are known to bind on the lysine tail of the histones, and then they drive the expression of these androgen driven genes. So, the idea was like if we use bromodomain inhibitor, we could attenuate the expression of DLX1, and that is what we did. We used these VCAP cells, treated these cells with JQ, which is a bromodomain inhibitor, and we can see there is a significant decrease in the expression of ERG followed by like DLX1, which is driving the expression of DLX1. And then when we perform this cell based assay, similar kind of effect was obtained with JQ1 treatment, and also with the anti-androgen along with JQ1, so significant decrease in cell proliferation as well as cell migration. Then finally, to recapitulate this entire mechanism in mice model, we implanted these cells in the mice, immunodeficient mice, and randomized them into 4 groups and treated them either with enza, enzalutamide alone, JQ, or a combination of enza and JQ, and you can see there is a significant decrease in the expression in the tumor burden of the mice which were administered with JQ or a combinatorial therapy with enza and JQ. And likewise, there was a significant reduction in the cells metastasized to bone marrow and lung in the mice treated with enza or JQ1 or enza and JQ1 both combined. So, this is a model that we propose. So, I did not talk about the ERG independent machinery, but here FOXC1 and AR and ARV7, they drive the expression of DLX1, whereas in the ERG positive patient is the 2 ERG and AR, they drive the expression. And also like these are the 2 target genes that we identified, LDH1 and HNF1. So, finally what we did here, we proposed the mechanism of DLX1 overexpression and also provided a therapeutic strategy how to target the patient who are having this DLX1 overexpression as well as ERG overexpression or negative. So, So again, like thanks to my collaborator within India as well as abroad, because without their like support, I would not able to achieve what I have presented here. And obviously, the fantastic people with whom I have worked. This is an old picture, so data I presented is like these students they already graduated, and this is a new group. And thanks to funding agency for funding the research that I do. Without that, it's not possible. Thanks.
Thank you.
Thanks, Bushra. I just had one question. Would you say that SPINK1 upregulation, which you, as you've shown, is mediated either through EZH2 mediated repression of the two MIRs, as well as AR resistance? Are these two mechanisms mutually exclusive, or do they cooperate?
I mean, I won't say mutually exclusive. There is some overlap because not all the patients. It's a heterogeneous disease. So in some patient, you see there is a operation of both mechanism through microRNA silencing as well as through ADT. And in some patient, you don't see. You see only one microRNA. For example, we see 421, but not the 338. So it's very heterogeneous.
Okay.
Yeah.
So in those tumors or in those patients where there is cooperation, is it a slightly more aggressive disease than the one in which you have just one mechanism operated?
So again, like we didn't classify patient like that cohort based on microRNA typing because it's difficult. Like we cannot do microRNA staining, so we don't know. But I assume because these are two tumor suppressor microRNA which have multiple targets. You see like in EMT and cancer stem cells. So probably it could be having much more aggressive disease.
Okay,
Yeah.
thank you.
Just a very nice talk. Just a clarificatory question. SPINK1 mutations are found very strongly associated in pancreatic cancer. In this case, do you not find any mutations either in the promoter or in the gene? You've gone directly to the microRNA. Are there no other explanations?
So again, like one of the mutations which are known for pancreatitis is N34S, right? So we looked for those mutations in the protease domain, but there were no such mutations. There are few mutations in the DNB, this mutation database, which are more into promoter region, but not in the main body, not in the protease region. So when we, in my postdoc lab, actually at that time we were thinking like maybe it's through mutation in the protease domain, and we generated some line which overexpressed the mutated version of the SPINK1. So it was behaving in the similar way as the wild type. So I think it's more of a protease independent activity of SPINK1.
But in the promoter region, don't you have mutations?
But they were not any like inactivating mutation. They were just passenger like with no significance. Like
no, in the promoter you would have underexpression or overexpression. That would
no. So they were not.
They were not.
No.
Okay.